animals as well as some non-human primates across 5, 6 completely uncorrelated disease areas where we tested the biology, and we have demonstrated in every case that this works, right? So to me, this is the future of medicine. This is programmed medicines. What we do is with our medicines, we enter the genome, the nucleus, like you said, the yolk, right? Home into the DNA, go to a very specific location that we designate in our medicine, and lay an epigenetic mark there that tunes a particular gene to resolve a particular disease in a particular cell type. So you give one therapeutic that lasts for that period. Now imagine the beauty of what I'm saying. So the mRNA is gone, the proteins that are expressed are gone. So think about it. There is no drug in your system left anymore, but the effect that we engineered stays. Right? Now think of that from drug-drug interaction, combination therapeutics. We literally have no drug in the system.
Hi, this is Liz Parrish and Sarah Vanan with the Buying Time podcast. Today we have a special guest, Mahesh Karande from Omega Therapeutics. We're going to find out what they do and how it's going to revolutionize your life.
Mahesh, over to you. Would you like to introduce yourself?
Sure. Hi, Liz and Saravanan. Thank you very much for having me on this podcast. So I'm Mahesh Karande. I'm the president and CEO of a biotech company situated in Cambridge, Massachusetts called Omega Therapeutics. And what we are doing is pioneering a completely new class of medicines where we are the first systematic use of mRNA therapeutics as programmable epigenetic medicines. And I would love to talk more about that, as we get into this discussion.
Mahesh, could I ask you first how you got started, how this became an interest to you, and how early in your life you got started, and how you came to the conclusion that this is what you want to do with your life at this time?
Yeah, that's a great question. I think it takes me back many, many years. So look, I'm an engineer by training, grew up in India, in Bombay, and I came to the United States to get a PhD in engineering. And this was way back, about close to 30 years ago. And as I was working through my PhD, I realized that, I was more interested sort of in the business aspects of things. So I actually stepped out, got a master's, stepped out, worked, and then applied to business school. And that sort of started me on my journey. And I, went to the Wharton School of Business in Philadelphia, University of Pennsylvania. And when I graduated, I happened to get an opportunity to work for a consulting firm. It's called McKinsey and Company. And when I worked at McKinsey, I started getting involved in telecom and technology, which obviously made sense, given that I was an engineer, but at the same time started working in pharmaceuticals and medical devices by the virtue of being in New Jersey, right? And that's the pharma capital of the world. And it was kind of serendipitous because, my family is in that business. My father worked for, Squibb in India all his life in very senior roles, and both my sisters are doctors. And I just happened to be an engineer who eventually stumbled into the pharmaceutical world. And, fast forwarding that, as I grew in the pharmaceutical industry, once, I left McKinsey and I joined eventually Novartis. And what I realized through my journey at Novartis is that it's an industry that allows you to do your work, everyday job, but at the same time, really gives back because you are all about developing innovative medicines to help patients. And, along my journey in Novartis, I was fortunate enough to work in emerging markets. I spent 3 years, actually 6, 7 years, 3 years in Switzerland and in Egypt and in Africa, really working on everything from Africa to Australia. And that gave me a sense of, an ethos that I developed that now is also my personal ethos and my company ethos, which is, I'll talk about the ethos in a second, but it focused me on, suffering and making sure that we are able to put patients first. And I think, as as human beings, as life expectancy increases, as populations grow, disease continues to grow. And there's going to be a continuing need to really solve that problem with more and more innovation, but at the same time making it affordable. Having thought that through, I realized that the chosen industry that I was in now, 20, 25 years ago, later, right, was the right place for me. And as I I think the next question is, how did I leave sort of the big pharmaceutical world and enter this extremely innovative world of biotech? So I'll tell you a little story about Omega. So about 5 years ago, when I was talking to the founders of Omega for a completely different reason, not really looking for a job. I already had a job. I was, working as a CEO of another startup developing antibiotics, gram-negative antibiotics. And when I was talking to this founder, what I realized is, and he was a venture capitalist, what I realized is that the science that he was talking about, if that could be translated into a drug development platform, it literally had the potential of changing medicine. And what it struck me in that conversation was that there's only once or twice in your life that you get an opportunity that can be that disruptive, that transformative, that when you're retired, when you're done with your life's work, you look back and think about those discontinuities that happened and that you were a part of it. That's what Omega sort of you know portended to be, and it was a very clear decision on my part then to actually join. And that's really how I stumbled into it. And I'm happy to tell you that 4 years after joining, we've taken this completely esoteric science, which we'll talk about later, and translated that into a true drug development platform. We are actually in the clinic with our first program in oncology. So
Great.
that's really the background story.
Now I want to know exactly what Omega Therapeutics does. And I want to know in a very, very simple way in which anyone could understand, including me.
That includes me too, Liz. Um, so let me try, right? Let me try and put it into very, very simple words. So look, I think, as the human body, um, you know, contains genes, right? And our DNA. And if you think there are about 20,000 odd genes in the human body, right? All of us have the same genes, right? And they essentially, the way life works is genes express, right? That means, there's so think of that as the hardware of the body, right? Genes and their regulatory elements and all of that, which is in every cell of the body, distributed across the 23 chromosomes, right? Every cell of the body, right? And the way life works is genes express, right? So the DNA, which is essentially that expresses, and it's a process, called transcription by itself, not so important to note, but a process called transcription by which mRNA is created. mRNA is basically the messenger RNA. So that takes the message from from the genes, right, and creates protein, right? So if you think of it, DNA or genes get transcribed to mRNA, that gets translated into protein, and protein, as we know, is essentially the building block of life, and that's how life goes on. And this is happening constantly.
Can I give an analogy for this for some of our
Yeah.
viewers who doesn't have the background? Please correct me if I'm wrong. So your body is made of many cells. I think last count was about 39, 40 trillion cells. And each cell, when you open, there is a nucleus inside. This is like exactly like there is a yellow yolk in the middle of an egg. And if you have cell with the egg, cell is actually one large, chicken egg is one large cell, and the yolk is the nucleus. And inside the nucleus, you have a lot of spindle-like structures. In human beings, I think we have like about 23 of them. And those spindles, you take them and they spun around. They're made of strings, and those strings are spun, and they look like the spindles. And if you take out those spindles, string them, stretch out the strings, we call them DNA. And that DNA is like a string. In that, there are messages encrypted using chemical molecules. And those encrypted segments is what we call as genes. Am I right in understanding this? I have two gene experts here.
I think, yes, I think your
Okay.
analogy is a good one.
and
Yes.
and then so when we talk about DNA, the way that I see it, I am a civil engineer. When we talk about DNA, the way that I see it is like a blueprint for you to build a house, and if your house is the body, and when you say that the proteins are made from mRNA, which are messenger RNAs, uh, so the DNA is a blueprint, and somebody reads the blueprint and goes and tells somebody else that, you know what, this is how you made the building block, make the building blocks, and building blocks of the body is the protein. You make the
Mhm.
protein and then go and start building the body.
You gave me a really good segue, Saravanan, because you said something tells the DNA, right, to go and build this, that, and the other, and that's what the building block of the protein creates, right? So that something that you're talking about is essentially the epigenome. So what you describe is called the genome, which is, if you think of, from a computer standpoint, the hardware of the body, right? It's in every cell and it's repeated in every cell of the body, right? That's something which tells it to go and do something, to build eventually the protein, right? Is the epigenome. Think of that as the operating system, right? Which is now telling that hardware what to do, right? So the way nature works is exactly like you described. So there is this, there's a hardware system, right? Which is all of the DNA, etc., that you talked about. And the epigenome is what controls it, right? So the way nature works, and Mother Nature is very, very smart. Mother Nature doesn't do it in ones or twos. It's a very systematic approach, right? So that is called epigenetics. That software, that operating system actually is called epigenetics, and that is what tells the DNA what to do, right? So what Omega Therapeutics actually has figured out, as we were looking at the organization of genes and their regulatory elements, what we realized is genes and the regulatory elements are very systematically organized in these three-dimensional loops, right? Even which form that strand within a single cell. And they're distributed across those 23 chromosomes, which is what you call spindles, right? And what tells that, what the way Mother Nature does it, it does something called laying or modifying epigenetic marks. So that's sort of a program that actually Mother Nature writes in that operating system, which actually tells that gene to express to a certain level, right? And then the whole process of mRNA to protein conversion happens and life goes on. And this is happening constantly as we are talking. Now, if you think about disease, how does disease happen? Disease happens if that program goes wrong somewhere. That means, the gene was supposed to express at this level, but it expressed at a completely different level, right? That's really how disease occurs. Now, if you had a way to say, well, this disease occurred, that means this gene got a wrong program. So let me figure out how to give it the right program. And if you're able to give it the right program and bring the gene's expression back to a normal range of expression, right, then you would resolve disease, right? Just by logic. That is exactly what Omega Therapeutics does. And this is the first time anybody is doing it. So what we are doing is we are using our completely new class of drugs. They are called epigenomic controllers because they control the epigenome, right? And essentially what they do is they go and lay epigenetic marks at the sites. These are regulatory sites which nature uses to tell the gene what to do, and we lay those marks at those sites that frankly allows us to control a gene. So think of a thermostat or a rheostat or even the regulator of a ceiling fan that you just sort of regulate to the right level. That's what we are able to do. So literally what we do is with our medicines, we enter the genome, the nucleus, like you said, the yolk, right? Home into the DNA, go to a very specific location that we designate in our medicine, and lay an epigenetic mark there that tunes a particular gene to resolve a particular disease in a particular cell type. So, what that means is, as I said, all of this exists in every cell. That means it's in the heart cell, it's in the lung cell. So, if you're targeting a disease in the lung, you want to go to the lung cell and give that message only to those cells. And that's what we do. Does that make sense? That's what Omega Therapeutics does.
Yeah, so I think like it just a short, brief way of explaining it. So you have your whole genome, and your genome is expressing for genes, and it's why your nose looks different than your toes because they're expressing in a way that is specific to certain tissue. There are genes in your genome that are methylated and unmethylated. So some of the genes are turned on for certain expressions to ensure that they're doing the right job, and some genes are turned off. As we age or we become diseased, specific genes may become turned on or turned off. For one example, if you go and drink a lot of alcohol, which I do not suggest, you will start to get protective genes that help your liver function. They'll actually try to help it protect it against the alcohol. Or if you're exposed to sun, you're obviously also expressing different genes. But what you guys are doing is you're going in and you're saying, here is a disease state, and we want to correct it or control it at the epigenome. And in other words, we want to either turn on or turn off specific genes. My question is how exact is this? When I started in 2011 working in the stem cell space, I did meet with a lot of people who were epigeneticists, meaning that they worked on controlling the genome from the outside. But at that time, they were having a problem because they could not control certain areas. So at that point, they were going after things like cancer, but they found when they turned on or turned off certain genes, it was actually affecting downstream effects.
So all of the work that you referenced, right, a lot, on the genome, right, and without getting into, by the way, Saravanan, for the sake of audience, without getting into sort of technical lingo, right? There were studies, they're called GWAS studies, they were done. And, like you explained, right? DNA is a string and genes sit along. That was the conventional knowledge, right? What we delineate, Omega Therapeutics was founded on science that was literally delineated in 2016 in a Cell paper. So it is that new. And it is, Liz, I suspect you might know what I'm talking about, but just to put sort of, a frame around it, right? Genes and DNA is not linear. It's actually these genes and their regulatory elements sit in these three-dimensional conserved loops that we call insulated genomic domains. And the regulators that control the genes sit only inside that insulated genomic domain, and any regulatory element that is outside it doesn't control the gene. So now you have these 15,000 IGDs that are spread across the 23 chromosomes that are ubiquitous in every cell. So first and foremost, right, what we do is we look at the IGD, which gives us a complete level of control on the gene, not just going to a promoter of the gene, but all of the other regulatory elements. So I think we have much more than one sort of handle to control the gene. And then the second thing is, you said something interesting, which is turning the gene on or turning the gene off. That is really looking at it from the old world or editing perspective, because that's what they do, on or off, mostly off, right? What we are doing is, because we use all of these regulators and we use epigenetics, right? And pre-transcriptionally, we tune the gene. So it's not 100% on or 100% off. We can tune the gene to the right level of expression, because in cancer, this is the problem, right? What happens in cancer is, if genes are turned on or highly expressed, right? If you try to bring them back to a normal expression at the mRNA level or the protein level or any other level, right? They auto-regulate and they crank more and more of the mRNA, more and more of the protein. And at some point, you can't put enough drug in the system. That's one piece. The second piece is exactly what you said, which is, if you control a particular gene, these are master regulatory genes and they have lots of other genes in their cascade, right? That means you have to control the cascade. That means you have to hit that gene specifically and tune it biologically only to a level where you're not going to disrupt the cascade. So there's a lot of science and art that goes into it, and that is what we do. So our specificity is exquisite. We are able to, I'm not showing you any slides today, but we have genome-wide maps where we have gone after a specific gene. So we can design our controller to do this. We decide on chromosome 8, on a particular IGD, there sits a gene, and I'm going to go and downregulate that or make an epigenetic change at its, one of its enhancers, right? And only there. And we actually have done genome wide plots where we have looked at the entire genome where the flux is below a certain line on a logarithmic scale, and we have been able to change 2 to 4 log only at that particular point that we targeted. So, and by the way, as we got this approved through the FDA, FDA asked us all of these questions, so we have sort of demonstrated this. So the specificity is super exquisite, and we have been able to achieve.
How are you able to ensure that other genes are not being modified as well?
How do you make the change itself? Now, both of you already know this, but I have not.
I'll address both these questions. I think Liz's question is actually a very important one, right? Because otherwise, if you have drugs that go and act everywhere, then you're not going to have the effect. So let me take that first. So, Liz, I think you hit upon the linchpin of specificity, which is the sequences of these regulatory elements. The interesting thing about the sequences of these regulatory elements. So you take one cell for a second, right? Just one cell, which has all of the chromosomes, 23, and these 15,000 IGDs that are distributed across those 23 chromosomes, right? Each of them contain 1 to 10 genes, on an average 3 genes. And that's how our genes are distributed, right? If you go to a single IGD, take any of those 15,000 and take any regulator out of that, the sequence of that regulator is unique in the entire genome, right? So now what that means is, let's say I targeted a regulator on IGD1, for example, right? Just let's give it a number, IGD1, right? And regulator A. Regulator A will be repeated in every cell of the body on that IGD, but none of the other regulators in those 15,000 IGDs, could be hundreds of thousands of them, will have the same sequence. That is something that we figured out. That's the smartness of Mother Nature. So that's one handle on specificity, right? Now, the second piece is what you actually said earlier, which is, look, and I said that these things are repeated in every cell of the body. So the same structure in the heart cell or the lung cell. So what that then means is, if you are targeting a lung disease or a heart disease, you first need to deliver the medicine to the right tissue, which is the lung tissue or the heart tissue. And once you deliver it to the right tissue, then it homes in into that particular gene, right? And Saravanan, I'll get to you how actually that happens and what we do there, right? Homes into that like a homing pigeon with a nucleotide sequence that is 21 bases long. Imagine you have 3+ billion bases in a cell, and we are targeting 21 bases. That's how specific we are, right? So you get the tissue specificity, and then you get the cellular specificity and the genome specificity, right? And that allows us not to have off-target effects. And we have demonstrated that. So I think this is where we are with the science. Now, to answer Saravanan's question, so the drug has two parts, right? One is what Liz was asking, which is a homing device. We call it the DNA binding domain that homes in and attaches to that particular sequence. And then the second piece of it, right, is an epigenomic effector. These are naturally occurring proteins that, like I said, Mother Nature uses, right, human proteins that either do things like methylation, demethylation, histone modification, acetylation. So these are all sort of epigenetic mechanisms. So we can choose that, right, that allows a tuning of the gene to a certain level for a duration. So Saravanan, think of it as a 2x2 matrix. On the y-axis, you have levels of up and down regulation, and on the x-axis, you have duration. So from a few days to a few weeks to a few months, right? Depending on the disease, we can choose which epigenomic effector or effectors that we want to actually use, right? And design those into our drug to have the effect that we want for the duration that we want. So, for example, for cancer, our drugs are effective with a single therapeutic at the beginning, are effective for 2 weeks, right? And then you get a drug holiday, like cancer drugs usually do. And then you again treat for 2 weeks, right? And for a chronic disease, we have effectors actually. We have data where with a single treatment, they last for 6 to 8 months. We already have demonstrated that. So that's how we do it. And let me get to the meat of your question, which is, I talked earlier about these drugs being, I don't want these to be some magical drugs. This is what we are delivering is mRNA. Now, everybody knows mRNA because of the vaccines, right? So mRNA is messenger RNA. But the beautiful thing about how we use mRNA is we use mRNA that codes for these two proteins. So the DNA binding domain is a protein and the effector is a protein. That's why I want to use the software analogy. So the DNA, the mRNA, sort of as it is injected, it goes homes into the right tissue, right? Like I said earlier, homes into the right cell type, finds its way through something called the ribosomal machinery into the nucleus of the cell. And once it's in the nucleus of the cell, it expresses these two proteins, which home into that specific location in the genome and make the epigenetic change there. So that's why these are mRNA therapeutics that are delivered into the particular cell type. Does that make sense?
Yeah, so mRNA is generally pretty short acting. How are you getting such long expression?
That's a beautiful question, Liz, and that's actually the beauty of a medicine, right? The mRNA expresses these two proteins. The mRNA is gone within a matter of few hours. These proteins, right? The proteins are gone within a matter of couple of days. Okay, but what stays behind is the epigenetic change we engineered, right? If we engineered, you talked about methylation. Methylation, is long acting. If we methylated a site, right, we know that site, and we have chosen an effector that will methylate a site. We know that it will act for 2 months or 3 months, depending on the what we chose, and that's it. So you give one therapeutic that lasts for that period. Now imagine the beauty of what I'm saying. So the mRNA is gone, the proteins that are expressed are gone. So think about it. There is no drug in your system left anymore, but the effect that we engineered stays. Now think of that from drug-drug interaction, combination therapeutics. We literally have no drug in the system.
Is this like on the lines of silencing mRNA, or this is different because this is actually affecting the genome?
Yeah, this is not silencing mRNA. This is basically, we are using mRNA as a means to an end. So again, remember, you have the DNA, you have the mRNA and the protein. We are acting at the DNA level. But what we are doing is using epigenetics, and the way we are getting to the epigenome or the genome is by using mRNA. So we get through the ribosomal machinery into the cell. That's the purpose of the mRNA. So we are not using mRNA as a therapeutic. We are using mRNA to express these two proteins, which became the therapeutic.
So the you have not a permanent effect, but you have a long-term effect because you're either demethylating or methylating the genome, which then the cell eventually responds by reverting back to the original, but
Eventually.
for a matter of weeks. I think that's really cool.
Eventually. It is cool. And Liz, by the way, right, look, I mean, we have just scratched the surface, right? Our first program is in oncology, which is we have designed for 2 weeks. Like I said, for chronic conditions, we have our, omega epigenomic controllers, we call them, that we have devised that work for 6 months plus, right? Think of this now. This is essentially the company's existence for 5.5 years, right? You know that Mother Nature, right, either turns on genes or turns them off or expresses them for long periods of time, putatively our entire life. That means we should be able to do that, right? So you're right. If we use methylation, demethylation, we also use other, histone modification, acetylation, all the other shorter acting mechanisms as well, right? And these mechanisms are handed down daughter cells as well, right? So our experiments that is 6-7 months long, these are in cell lines that divide every 24 hours. So you're talking about generations of cell lines. Now the beauty of this is it will taper off at some point, and then you can give another treatment, right? Now, who is to say that this is temporary? This could be permanent depending on what is permanent. I mean, if we can do it for 1 year, if we can do it for 2 years, is that good? So the sky is the limit here.
So actually, this is probably a nice pre-tester for somatic cell gene therapy or CRISPR editing or TALEN editing. And what I mean by that is that if for the disease state you actually need a super long-term effect, you're a great testing mechanism to go and find out if that's true. So for instance, if a person needed to redose continually because they needed this effect constantly, you could then just go in and edit the genome.
I would not do that at all. I think let's think of it a little bit differently, right? So if you think of drug development, right? you know, most of our lives we have had small molecules which act on the protein and large molecules, right? And then, in the last decade or so, mRNA medicines came where they said, well, let me actually tackle the mRNA, right? And those work, right, in vaccines, etc., and some rare diseases here and there, right? Some drugs have been approved with siRNAs, but they have not worked in things like oncology, etc., for various reasons. For example, autoregulation, that I talked about.
Mhm.
Then came the gene therapy and gene editing companies that took aim directly at the DNA and started making wholesale nucleic acid sequence changes. These are phenomenal advances in medicine, no doubt about it. But imagine the collateral risk that they carry. None of these drugs are approved without 20-year follow-ups. Why? Because we don't know what is happening when you edit stuff, right? So the way to really think about it, what is the next level? The next best thing is what we are doing, which is we are making no nucleic acid sequence changes. We are literally going to the mechanisms that Mother Nature uses. It's akin to, let me give you an analogy. Okay, if you are basically in a room full of light bulbs and circuits, okay, if a light bulb goes off, gene editing will go and replace that light bulb. I mean, gene therapy. What they will do is like, hey, this light bulb doesn't work. Let me just put a new light bulb and figure out what else happens. Okay, that will work only if the light bulb itself is wrong, right? From a gene therapy standpoint, if you don't have a gene, if you don't have a functional working copy of a gene, you are left with nothing but gene therapy. So I absolutely agree, right? But think about how rare that is. Okay, so that's the light bulb analogy for gene therapy. Gene editing is like, hey, this bulb is not working. There's something wrong in this wiring. Let me just cut this wiring and I'll put just a completely new wiring, right? And connect it back into a different socket. So when you turn this off, this bulb turns off because I have this other switch which turns this one on, right? That's great, but now you've completely mangled the integrity of the network, electrical network that you have, right? Now in electrical networks it's not as problematic in our room because we get the light, but now think of that as the human body, that you went and just snipped something, you cut something out, you have no idea where else it is being used, right? So what we are doing is we are saying, look, there's something wrong in this wiring, right? What is wrong in this wiring? Let me go and actually fix that, right? And make that system work correctly, because there's something going on that's not working correctly. That's what we are doing. So if you think, we are making no nucleic acid sequence changes, and we are giving therapy which could be chronic, which could be for short term, across every gene, because what I said controls every gene in the body. That means it controls every disease. So I would not at all look at this as a precursor to say, okay, wow, now I can go and edit this. In fact, I would say, do I really need editing anymore? Because that is problematic.
Yeah, I suppose that in the long run, it would depend on the cost of the drug and the use for the patient. But yes, okay, I think that that's fair.
What kind of results are you seeing now? Mahesh, you said you have something in oncology therapy.
Yeah. So look, I mean, I think, I cannot actually talk about our clinical work right now because we're a public company and we haven't talked publicly about it. So I can't talk about our results in the clinic. But when they come out, Saravanan, I'm happy to talk then. But, the way to think of it, right? We have a huge body of preclinical work, right, that we have done. That means on species other than humans. And it shows tremendous results, which formed the basis of the FDA, the regulatory body, approving our drug to be treated in the clinic, right? So, and those results have been fantastic. And that's why we feel that this will work well in the clinic. And by the way, there's one more thing that I actually forgot to mention, which gives us confidence about this working in the clinic. So, I talked about this system, right? These IGDs and this entire system. The interesting thing is that these IGDs are evolutionarily conserved. What that means is, first of all, within species, all of us have the exact same IGDs and this exact same genome, right? Funnily enough, that these IGDs are actually conserved in all mammalian species. Now, in lower order mammalian species, you will have the same structures of these IGDs and the genes sitting in them, but the sequences of the regulatory elements that Liz was talking about earlier are different, right? But when you come to non human primates and humans, the IGD system is pretty much intact. So it's the same in non-human primates as in humans. So one thing, to think about that we are actually going to prove out, hopefully, is that what you are going to see in non-human primates will give you a pretty good handle on what you will see in humans. Of course, the biological systems are different, so we have to demonstrate that in the clinic in humans. But the biology is conserved. So when you're going after such fundamental biology, you know that the biology is going to work.
I think that would lead into my next question. Outside of non-human primates, how predictive do you think that the other animal models are? Because there's a lot of argument that that's why so many drugs have failed in clinical trials, and that companies are spending millions of dollars, tens of millions, and sometimes hundreds of millions of dollars on animals that are non-predictive of how a drug.
Absolutely. And look, I think, Liz, the reason that happens and that has happened, right? And look, frankly, as AI, etc., takes, more better footing, right? We will eventually be able to, go away from these, clinical trials in animals, etc. Those days are yet to come, right? While those days are yet to come, the way regulations work is that you kind of need to try this in lower order species before you put them in humans, because human life is extremely valuable, which makes sense, right? From looking at it from that viewpoint. So now, the reason drugs have failed, I believe, is because exactly what you said, that the animal systems are sometimes not predictive of human systems, right? Fair enough. If you're thinking about small molecules, large molecules, even editing potentially, absolutely. That's why you have probabilities of success that are that low, right? One of the things that we are doing is we are going after such conserved fundamental biology, and we are not sort of using, the entire humans. If you think of the central dogma, right? Downstream in the central dogma, you are using biological processes that could be very different between all the animal species. What we are doing is we are going and making epigenetic changes, right? That simply tune the gene at the genome level, right, in conserved biology. So think of it this way: you have a gene expressing to a certain level of mRNA is produced, certain level of protein is produced, right? Disease happens because either that is excessive or it is less, right? If you are able to pre-transcriptionally bring the gene back to a normal level of expression, then you would imagine that the mRNA created and the protein created is at the normal level, right? That means the biological system should work normally. So I do believe that the probability of success for a therapy like ours is going to be extremely high. And we have tested this, right? So look, even gene therapy, cell therapy, editing have probabilities of success that are better than small molecules and large molecules. We believe that our probabilities of success are actually going to be even higher. And I'll tell you one thing: we have tested this across, we have in vivo data, animal data, right? Through animals as well as some non-human primates, across 5 6 completely uncorrelated disease areas where we tested the biology, and we have demonstrated in every case that this works, right? So to me, this is the future of medicine. This is programmed medicines. So what we are doing is, this is the era of programmable medicine. So we are telling medicines what to do and where to act, as opposed to throwing in a small molecule that goes everywhere in the body and does whatever it does across everything it encounters, and also in the tissues that it's necessary to do. And that's why drugs work. We are programming medicines to go and do very specific things by using very specific biology. This is a completely different paradigm of the way we are thinking today, right? Given what is in existence, and we believe that these therapies, as they come more and more, they become more and more common, right? We are the pioneers. We're the first, actually, that you will eventually even have conversations at the regulator level where you know regulatory pathways need to be changed. We are literally at the cutting edge of it.
Yes, I agree with that, because the regulatory pathways were really established, and the difficulty and the high cost and the long time to get drugs to patient were established because of all of those off-target effects of small molecules.
Absolutely, absolutely. Liz, the long cost, the high cost. I'll give you an example. So, the drug that we have put into the clinic now, okay, from scratch, where we first looked at the gene and the IGD, designed it, tested it, did the IND enabling work, got the IND cleared. You know how much time it took me? 27 months.
Wow.
Industry best is 4.5 years. Now, we are doing this in 27 months. So, you know, as long as we can deliver to the tissue. So, if you think about where the industry sort of pain point or the bottleneck is right now, is delivery to tissues, right? We are using lipid nanoparticles. People are using AAV vectors. So, as you unlock more, so our first program is in the liver. Our second program we are working on is in the lung. We can deliver to those. So, liver, because we already knew how to deliver to the liver, it took us 27 months to clear an IND. That is unheard of.
That was what I was going to ask you next. We use AAV and then we're working on CMV, but CMV is a DNA-based vector. But I was wondering, how are you getting these in? Because, with CMV, we're looking at redosing, and that's really important with us. AAV, you can't do that. So in gene therapy, just so people know, we talk about serotypes and tropism. And serotype is a specific therapeutic designed for a tropism. So the serotype is designed to target a certain tissue. How are you creating serotypes to target specific tissues? In other words, how are you making a drug to only target the lungs, to only target the
As such, as a company, we are delivery modality agnostic. Okay. But in our initial work, our first few programs, we are using lipid nanoparticles. Now, lipid nanoparticles, as you know, mRNA and lipid nanoparticles, were made famous by the Moderna and the Pfizer vaccines. They are a modality that has been tested in, I think, more patients worldwide than every other biotech modality combined. So I think there's a plethora of safety information on that front. So we feel pretty good about it. So now, getting to your question, the chemical space, the SARs, right, for creating tropism to different tissue types in the LNP is such a vast area that not only us, but there are so many companies that are working on this. And these, I believe, are engineering problems. Saravanan, you and I being engineers, we know that engineering problems can be solved, right? Biological problems present themselves, and unless we understand them, there's no two ways about it, right? Engineering problems can be solved because we can create things that address engineering problems. To me, delivery to tissues through with LNP, for example, is an engineering or a chemical problem that we are solving. So omega, for example, internally, our own LNPs we can deliver to the lung. We have demonstrated that we can deliver to many other tissues as well. Now we are optimizing all of that. We are at the cutting edge of the industry in terms of delivering. And our second program, which we have declared, is in the lung for lung cancer, is the first of its kind using mRNA therapeutics, delivering it systemically. So, Liz, to answer your question in a short way, like you are working with CRVs, right, and trying to modify them so you get tissue specificity, and that there's no immunogenicity, etc. Immunogenicity doesn't exist much with LNPs, but we are decorating the chemical space of the LNP to be tissue tropic, and we have been able to achieve that, and that work will continue.
right. I think that the big burden of LMP, depending on what you're working with, is hepatotoxicity and the PEG reaction that probably was responsible for some of the blood clotting. So, there is susceptibility for patients to build an immune response even to the LMPs. How would you deliver to the whole body and avoid the hepatotoxicity?
LNP world has matured so much. So I think the early LNPs where we saw all of that, I think we are like, I don't know how many generations later in terms of LNP. Our LNPs are not necessarily, without getting into the details of it, pegylation is not necessary, right? You can do a lot of other things, right? We haven't seen any toxicity, hepatotoxicity beyond a certain level that is acceptable, right, with LNPs at all. And these other LNPs that we are devising, yeah, eventually they will go into the liver, right? Some of them, but let's say if a tissue targeting LNP is developed, a different tissue than lung, a majority, a good amount of therapeutic dose will go into that tissue first before eventually finding its way to the liver. But these are not hepatotoxic to beyond normal levels. So I think the industry has learned on that front, and these days the LNPs that we are working on are way safer than the ones that you are referring to.
What diseases they are looking to target over a period of next 2, 5, 10 year horizon? How we can repurpose it for longevity space?
At a simple level, we are able to target any gene and any insulated genomic domain that it sits in, or multiple genes, right, that sit in, an IGD. The funny thing is, when nature files multiple genes in a single IGD, they usually act along the same disease pathway because nature is smart. And the reason this is important is because that means we can target sets of genes or single genes with a single therapeutic. And Saravanan, what that means really is, to your question, is because we can pretty much target any gene, that means we can pretty much target any disease, right? So as we have looked through Omega's ability to target disease processes, think about any disease processes, neoplasia, which is cancer. You think of metabolics, you think of inflammation, you think of immunological pathways, you think of neurological pathways. We can target anything, and we have demonstrated this, at a cellular level, as well as some in vivo work across all of these diseases. So the broad spectrum of diseases is available as the technology matures more and more. Now, coming back to what Omega can do, in the short term, is as we unlock these diseases, right? Think of it this way: cancer is something I talked about, right? Because cancer happens to be a poster child for epigenetics. Cancer occurs most of the time because of an epigenetic change that happened that actually triggered the gene overexpression. Okay, so we can definitely go after cancer. Now, something that actually segues well into longevity is this second part that I'm talking about, right? Which is, what these IGDs control is not just gene expression, but they control cellular programming, right? Because genes express, that means cells are created, right? So think of degenerative disease, right? Think of a disease where your liver is degenerated because of alcoholic abuse, or maybe a viral insult. Doesn't matter what the etiology was. But let's say you're on the path of liver degeneration, right? What if you could target a gene which is a master regulator of hepatocyte or liver cell function, and turn that on so that you can create hepatocytes? We have actually done that. We have literally what we have done is, in a mouse liver, we have killed all the mouse liver cells and injected a basic human liver cell, and then actually tuned it up and created an entire mouse liver, humanized liver inside a mouse liver exoskeleton. So we can actually regenerate liver. So now that sort of, so cancer is uncontrolled cell growth, which we can control, right? Regenerative medicine is controlled cell growth, which we can engender.
All your Parkinson's and other neurodegenerative diseases, your dementia, maybe Alzheimer, these are all these things should be possible for it to fix.
We should be able to do that. So over time, right? I think the question will be whether we can deliver to those specific cells and tissues.
And I'm presuming that your infections and all are not your targets, because anyway, they are bacterial infections and viral infections anyway, because they have good solutions already available. That's not
Exactly,
something that,
exactly,
yeah.
exactly, right? I mean, so the primary source, if it is bacterial viral infection, right, genomic medicine is not the solution. But those bacterial viral infections lead to other things. So you can actually have cancer because of a viral infection. And whether it was, in liver cancer, for example, if liver cancer happens, right, and there's a gene called MYC, for this is the only technical piece I'll say here. It's a MYC gene, which is a master regulatory oncogene, which gets overexpressed in liver cancer. Okay, that liver cancer can happen because of a person drinking too much alcohol, or it can happen because of a hepatic virus that infected the person a long time ago, and the liver slowly got destroyed and eventually became cancerous. No matter what, we can go and fix it, because all of that tuning of the gene has to be done at the IGD level. So we won't treat the primary source of that bacteria virus, but we'll treat what it does.
Okay, so HPV is something that could become less of a threat in the coming future. Many such viral
Yeah.
infections could. Excellent. Liz, can you think of some way by which we can take this technology and adapt it for our longevity space?
Well, of course, what comes to my mind is obviously I'm very excited about this technology. I think it's just such an honor to meet Mahesh. I want to follow the technology. I want to follow the company. I would love to work with the company in the longevity space. My big question to longevity is that when we look at creating longer and healthier lifespans, so organ regeneration, that's huge. That's going to be massive. But we are going to actually have to repair a lot of damage, and we target the hallmarks of aging. So in a therapeutic like this, would a person be able to take a cocktail? So literally a cocktail, because we wouldn't want to just run towards neurodegeneration and neuroplasticity. We'd need to work on the liver, the brain, the whole body muscular system, the telomeres, the mitochondrial dysfunction, and of course protect against genomic stability. How do you foresee your therapeutics being able to do that in the future?
Yeah. So, Liz, I mean, I think, speaking about the future and from a, you know, somewhat a theoretical standpoint, right? Our platform can be used sort of with a single therapeutic. You can multiplex to various different IGDs and regions, right? Because all you're really doing is, inside an LNP packing, you can pack up to 8 to 10 mRNAs, right? mRNA constructs. Each of them code for, let's say different genes and different IGDs. And if you send it systemically in the body, right, it will use the mechanism where the mRNA will escape from the LNP and then home into the different parts of the body. So everything that you're saying theoretically is possible. I think obviously it will take a lot of time to get there, right? So from a technology standpoint, I don't see at all an issue. I think the question will be, how do you develop it for that, right? That is always the question. It takes time, it takes effort, it takes money, but eventually I think this solution can work there. Right now, having said that, I think you can take things one at a time. You can look at sort of effects of aging and go after some of those effects of aging first, right? As opposed to like a final cocktail that solves for everything. You can sort of prioritize things and say, okay, this is what I think we can work on. I mean, we are looking at systemic therapies where, you know, you can sort of, do damage repair at the DNA level, right? That's not one of our priorities right now, because in the early stage company, what our priority is to prove out the technology in something that is tangible, feasible, that actually makes sense to people, and then over time grow the company, right? If you think 20 years from today, I think, or even earlier, all this is possible. I think even prevention is possible, because if you know that certain genes are going to, in rare diseases, you know that in your adolescent age, you have the gene or the markers for, let's say, Friedreich's ataxia, which is sets in, in adolescent age. If I know that from a genetic test already for a child, why wouldn't I start treating that child with a therapy, when the child is 9 or 10 years old? We don't have regulatory pathways for that today, but 20 years from today, we might actually, right? So the world is a oyster, is how I would like to think of our platform, really.
Since you've brought up age, so you bring up children, and my desire is that we do not leave our children with the same diseases that we die of. But there is also the other side, that we may be able to save a whole generation of people with the technology now. How has your age played into your different views on what you do?
I mean, I think other than the fact that I just matured with age, and I think, my thinking changed, which I outlined early on, right? Which is, as I got more and more experience, as I experienced different parts of the world, fortunate to have traveled to over 75 countries and lived in, five or six of them. Um, it gave me a perspective on what's important in terms of innovating for medicines and, kinds of diseases to go after, and that eventually I would like to sort of create medicines that are, accessible to the entire world population, right? Beyond that, I don't think, if your question is, has my age sort of guided me to certain specific diseases? It hasn't. Fortunately, I live a pretty healthy life, right? And I'm working obviously with Amura on trying to make it even healthier. But besides that, but having said that, look, I think all of us get afflicted by disease. I think cancer is something that touches all of us. So I think solving some of the cancers is a high priority. Some of the diseases like Parkinson's, etc., hit home close to me, my colleagues, many friends, right? So I think, yeah, there is a you know list of diseases that I would love to tackle as the technology progresses, just because of what I've experienced. But that's kind of how I've been thinking about myself.
There is a disease that Liz and I are focused on treating, and then billions of people are afflicted by it. It's called aging. We wish that somebody takes it up seriously and works on it. What, Liz? What do you say?
Aging is the biggest driving factor for mortality, and so of course that's why we are focused on that. We do believe that we could target the most amount of diseases by affecting the mechanisms of aging and helping people get chronologically old but not biologically old.
Liz, no, I agree. Look, I think aging is an interesting thing, right? Because I think, and you guys are way ahead in terms of your thinking on aging than I am, because I'm never squarely focused on that. But to me, at least the way I think of it is, aging is a plethora of conditions that actually happen. Now, if there is a particular thing that we figure out that, look, this is sort of the mechanism or the pathway or the gene or set of genes that if we are able to really affect in a positive way, then you take care of the multiple pathways that lead to degeneration, which is a artifact of aging, right? That's a cool way to think of it. I just don't know yet whether that has been delineated, right? And I think it's important, the work that you guys are doing, because that continually will help shed more light on it. And if that gets delineated, then we know that we can do that. Because you think of any disease today, 50 years ago we didn't know those pathways, right? But now we know those pathways. So to me, it's important. So the work you do makes a lot of sense to me. And if we figure out where actually to act on certain pathways, I think we could do that.
Yeah, I believe it wasn't until the 1960s that the US government decided that cancer was a disease. It was just a process of aging before that. And because of that, people lived decades after diagnosis. I want to thank you so much for your work. We definitely are out of time. We look forward to having you back and talking about your company's successes as one after another happens. Thank you so much for joining us.
Yes, Liz. Thank you. Thank you for taking the time, inviting me.
Thanks, Mahesh. Thanks a lot for coming.